So my present study is focused on a cluster of microRNA named DLK-DIO3, and it can regulate the mitochondrial function in the dystrophic muscle in Duchenne muscular dystrophy. So especially for the audience of this meeting, and also we have already very comprehensive introduction about Duchenne. I just briefly mentioned that. So it's a rare genetic disease due to the mutation in the dystrophin gene. It's important for the attachment of the sarcolemma during the repeated cycle of contraction and relaxation. And with the absence of the protein, the muscle is subjected to a progressive muscle degeneration and weakness. And although there's many advancement in gene therapy approach, especially in the animal model, there's still no curative treatment at the moment for the DMD patient. So in our recent study, so we did a microRNA profiling in different model of Duchenne. So here we have the Duchenne patient serum. So we did the microRNA profiling in the serum, and here the in the DMD patient is in the GIMD. So this is the dog model of the DMD, and here's the mouse model. So in the three model, it appears to us that many microRNA in the same dynamic locus. I'm gonna briefly introduce afterward. It's called DLK-DIO3 microRNA, which I mentioned DD in short. So it's labeled in red here. So you can see in this volcano plot, there are many microRNA from this locus that are upregulated in the serum of the DMD patient and in the dog serum. And also the dysregulation of the DD-miRNA also found in the muscle biopsy of the same subject. So we see in the muscle biopsy of the DMD patient, GIMD dog, and also in the MDX mice. So we have very high upregulation of this locus in the three models that we study. So what is the locus about? So DLK-DIO3 locus is an imprinted locus. So it means that paternal chromosome and maternal chromosome are inherited differently. So we put our focus in the maternally inherited chromosome, where we have the expression of many non-coding RNA, especially the microRNA. So here is so this locus is a home for the largest microRNA cluster in the human genome with more than 50 microRNA. And the function of this locus is very well known and well characterized in stem cell field, especially in the embryonic and adult stem cell. So there are several papers also mentioned about muscle stem cell. It's also function in aging, and in the muscle phenotype, one of the most well-known phenotype for this locus is a calypic hypertrophy phenotype that we see in sheep. And we would like to check the expression level of the DD-miRNA during the regeneration process. So first of all, in vivo assay was done by fact sorting the satellite cell and let them in culture for activation and differentiation. And you can see that the distillation already seen in the satellite cell stage. So this is not something that is surprising because the locus play a role in the stemness of the stem cell. But the thing that are interesting us is that during the differentiation, the expression is raising up, and especially high, raising up higher in the MDX differentiated myotube. And the same pattern was seen in the model of acute regeneration muscle, where the muscle was injected and injury by the cardiotoxin, and you can see that the expression level was raising up in the day 10 to day 14, which is overlap with the time where we have newly formed muscle fiber. So it's success that the repeated regeneration process that are give to the operation of the locus, what we see in the dystrophic muscle. So let me put a small recap in this part, is that we identify a very high distillation of the microRNA DD locus in the DMD patient and animal model. And we suspect that the distillation is due to the regeneration process in the dystrophic muscle. And the next question we would like to ask is the mechanistic explanation for the operation. And we come back to the regulation of the locus. So the expression of the non-coding RNA, for example, the microRNA in the locus is mainly controlled by two regulatory sequence. It's called IgDMR and MET3DMR. So this two region is subjected to a lot of modification at epigenome, so especially DNA methylation and histone modification. And what we found is that corresponding to the upregulation of the locus, we have a reduction of the repressive marker, for example, the repressive histone marker H3K27 trimethine, and also we have a reduction in DNA methylation in one of the most important regulatory sequence, IgDMR. And so we conclude that the DD-miRNA overexpression is due to the epigenetic dysregulation at both level of DNA methylation and histone modification. So in order to study the function and the consequence of the operation of the DD-miRNA in the muscle, we have to select. We have put a selection here because there's more than 50 microRNA in this locus, and we cannot study all of them. So what we did is we tried to put some selection criteria, and we select 15 of them of interest and put the sequence of all of them in the AAV vector, which is a very useful tool for gene transfer in the muscle in vivo. And we injected them intramuscularly in the TA muscle of the wild type control. And there are two conditions to compare here: the mere condition containing 14 microRNA that are overexpressed at the same time, and the control group with no microRNA sequence. And here's the validation of the overexpression in vivo that we see. So we confirm that we are able to overexpress the microRNA in vivo via the AAV vector. And it's noteworthy that the overexpression level is similar with what we see in the dystrophic muscle of the mdx. And it's the thing that's surprising us at first is that with the overexpression of the DD-miRNA, this alter very severely the muscle function and histology. So you can see that the weight of the muscle that are reduced dramatically. So you see that 1/3 of the muscle weight was gone either in the 1 month after injection or 3 months after injection. At the histology level, because remember that we put the AAV vector with the transgene of GFP as a selective marker, and we inject intramuscularly, so it's give a lot of central nucleation in the control group, but with the presence of the microRNA, somehow the central nucleation, the percentage of central nucleated fiber was reduced more than half. And the fiber size distribution also shifted accordingly to the size and to the weight of the muscle. So here we have two conditions where we have the overexpression of the DD-miRNA. So first in the dystrophic muscle where we have the upregulation of the DD-miRNA, and we also have another condition where we artificially overexpress the microRNA in the muscle. And we want to compare between the two. So we did the transcriptome in these two conditions. And here is all the dysregulated genes that are found. So here you see that about around 2,000 genes that are found to be dysregulated in the DD-miRNA overexpressed muscle. And out of those, more than 75 of them that are commonly found with the dystrophic muscle. So when we plot the 1,500 gene to compare between the so this is the commonly dysregulated gene in the two conditions, and when we plot them, it's give a very high similarity or very high correlation between the two data sets. And it's also appeared to us that there's many mitochondria genes that are found to be downregulated in between the two conditions. And to get a more statistical mean for our analysis, we apply gene state enrichment analysis in our study. So what we found is that, and also because of the nature of the microRNA is to inhibit and to repress the gene expression. So that's why we put our focus in the downregulation part. So you can see that in the MDX muscle, there's many metabolic pathways that are found to be significantly downregulated in the dystrophic condition. And the downregulation here can somehow be recapitulated with the presence of the microRNA itself. So you see there are 7 of those, 7 metabolic pathways that are found to be similarly dysregulated with the presence of microRNA compared to the one in the dystrophic muscle. And but zoom in a bit for the only we focus about the mitochondria here. So here is a list of more than 1,000 genes of the mitochondria that are taken from mitochondria. And you can see that here is all the mitochondria genes that are dysregulated in the MDX, and here is all the mitochondria genes that are dysregulated in the DD-miRNA overexpressed muscle. So you see that, just focus in this part. So our 265 mitochondria genes that are dysregulated in the DD-miRNA muscle, 250 of those, so more than 95%, that are also commonly dysregulated in the dystrophic muscle. So it indicates that very high similarity between this condition. And we apply some functional tests. For example, we have the cytosynthase activity where we see, and mitochondria, we see a reduction in both two conditions. Here the electron transport chain also seem to be a target in MDX and DD-miRNA muscle. So here's just a visualization of the gene set enrichment analysis. So we see a reduction in at the transcriptome level, and at the activity level, we also found a loss of dysregulation in the dystrophic muscle and in the muscle with the presence of DD-miRNA. So just to conclude what I just presenting, so we identify a group of microRNA from DLK-1 DLK-3 locus are highly dysregulated in the Duchenne patient and animal model, and this dysregulation is due to the epigenetic dysregulation. And we also provide evidence that the DD-miRNA govern many metabolic pathways in the dystrophic muscle, in particular electron transport chain. So this give a very general overview. So in the dystrophic muscle, there are many in the dystrophic fiber. So we have the leakage in the sarcolemma because of the absence of the dystrophin gene, and so the calcium is get through. So the calcium get through, and the mitochondria is the function to buffering the calcium in the calcium intracellularly. And over time, the mitochondria become defective. So that's a commonly explanation for the mitochondrial deficiency in the dystrophic muscle. So in our recent study, was published earlier this year, we identified a cascade where one of the microRNA, so MBA379, this is also one of the microRNA from the DD-miRNA locus. And these microRNA that are responsible for a cascade that are controlling the expression of lipid protein, which is a key component in the complex 5, and this lead to the defective mitochondria. So it's in this present study, it's a generalization of all the microRNA from the locus, so way more microRNA than only one single microRNA here. So microRNA 379 is part of this, and we identify many targets of this microRNA in the mitochondria. So it's suggesting an alternative explanation or alternative mechanism explain the mitochondrial deficiency in the dystrophic muscle. And just to finish, I would like to acknowledge all the people that helping me out during the study, especially the progressive muscular dystrophy team, the Imaging platform, histology, animal facility, and better core. I would like to give a special thank to Marco Spinazzi for his insight and contribution. And I would like to thank to all the Zenithon and the funding. And thank you very much for your attention. I think that I only have a 20 minutes of presentation, so I'm not prepared for long.
Thank you very much. Hong or Hong only. Yeah. We extended your presentation time to hoping that now the discussion will cover the 3 previous speakers, or at least the 2 previous speakers. And so I invite Professor Matsuda to still here, hopefully.
Yeah.
He disappeared?
No, I'm here.
You are here. If the speaker present the last slide, please.
You're talking to me.
The previous slide.
Okay. Yeah.
This seems to bind the calcium, phosphorus, and mutation.
Yeah.
It is too easy, too simple interpretation, or this could be a topic of common discussion between people with different backgrounds.
Yes. So this is a question for me. You mean? Sorry, I don't get the question. Is it a question for me? Or?
My question is someone who presented, if Professor Matsuda recognized in this slide something that is relevant in his experiments.
Okay, so the question for the Professor Matsuda.
This is more a question for Professor Matsuda, but main time also for you.
Actually, we are aware of that. I think that the mitochondrial deficiency is, of course, due to the calcium uptake, either overcharge of calcium in the mitochondria. But we are suggesting it's not the only mechanism in dystrophic muscle. So we are providing the evidence for the alternative explanation for that. So that's what I trying to convince you and to convince the audience here.
That there is a common mechanism when there is nuclear altered transcription or the external factor of calcium entry.
That I don't really know.
At the end, they have the same general effect. They damage mitochondria. But they interfere, or are just 2 different examples of mechanisms who damage mitochondria.
Actually, we don't really know why. I mean, I provide information about the epigenetic change, but actually we cannot really know why the locus that are going up. We suspect that because of the regeneration process. So the regeneration process of course, the regeneration process give the change in the transcriptomic level, and maybe the calcium is one of the contribution factor for the contributing factor for the transcriptomic change. So that's what we think. But the bottom line here is that so the calcium overcharge is like physical mechanism for the mitochondrial dysfunction, and the mitochondrial dysfunction due to microRNA is like more about genetic cause. So it's like a cascade of cascade of molecular change in so the microRNA that are target the gene that are related to mitochondrial metabolism and lead to the metabolism defect. So it's like more about the genetic cause than the physical cause.
Thank you.
Hi, Hugo. Do I have time to ask a question?
Yes.
There is more time because I did not send the message to Hong that his presentation was extended, and therefore he was very, very precise in his time.
9 minutes.
You also extended my presentation on Saturday, I think, without telling me. So I'm adding slides right now. So I enjoyed your presentation, and I was wondering, about this. You're showing this cartoon in which I see calcium entry and the mitochondrial damage.
Yeah.
So the calcium entry pathway that you see being deleterious on mitochondria, is this through which?
Corrado.
Do you know? Did you check? So you think you have what you activate is a store-operated calcium entry, or simply is the damage through the membrane?
So the question is more about the calcium. More about the calcium, you mean? More about the calcium.
I'm looking at this cartoon I see over here.
Yeah.
And I see a calcium entering pathway from the outside.
Yes.
I see also obviously calcium coming in through the damage of the membrane,
Yeah.
The membrane is really damaged. I think that it has been shown by several groups that calcium is upregulated it's higher at rest in the muscle fibers. Do you think there is a specific calcium entry pathway which is activated that deregulates the calcium levels in the fibers in muscular dystrophy?
I think probably yes, but I don't really know about this issue, so I would say yes, but I believe it's yes. But to be honest, I don't really know much about the story about the calcium defect in Duchenne.
There are, if I can say, there are delta legion. And I would like to ask, have you studied any other microRNA like, for instance, MIR207, 206, MIR1, that are known to be high in Duchenne dystrophy? MIR206 microRNA.
Yeah. Sorry, I'm not catching the question. Is this the question?
Yeah.
So can you repeat the question, please?
What about what are called the microRNA, like MIR206?
Yes. Your question is what happened with the microRNA 206, or?
Yeah.
So the 206, I think, is important. It's a myomia. So the pattern of the dispersion we see here is totally different, because when you have a leakage in the membrane of the muscle fiber in the dystrophic muscle, the microRNA was just getting out kind of passively in the serum. So we see higher level in the serum of the MIR1 or MIR206. But in this case,
yes, but your MIR are also overexpressed, or not?
Yeah. Yes, our MIRNA is overexpressed in both serum and in the muscle. So of course we are not really understanding. We are not conclude that the muscle is the only contribution for the upregulation in the serum, but we suspect that we believe that the muscle is the main contributor here for the upregulation in the serum.
Can I add something here? just to complete, the myomere, in fact, are upregulated very strongly in the serum of dystrophic animal and human, but in the muscle, most of the myomere, in fact, are downregulated. And this group of miRNAs that we identified, DLK-DIO3, are among the most highly upregulated in Duchenne, and by the way, not only in Duchenne, other muscular dystrophies. So while there's a lot of studies on the myomere, which developmentally highly important, in terms of repression of gene expression in some dystrophic muscle, probably the DLK-DIO3 miRNA are more important than the myomeres. So I don't know if you hear me very well. Can you hear me?
Yeah.
So just to
So what you are saying is that the myomeres are opposite in dystrophic muscle,
Yes.
low in muscle, high in serum, and here they are both up.
Exactly.
Yeah. Also, concerning the calcium question of before, so we are not specialized in calcium toxification of the mitochondria. It's just a common vision to describe. There's somebody's microphone. Whose microphone is that?
There was some noise. Anyhow, maybe it is time to move to the next speaker that will talk about something for me related. Maybe it's not true, but I hope to learn.